Welcome to the BioInsights, where science comes alive.
Today, we're uncovering how a microscopic survival tactic, something totally invisible to the naked eye, literally shapes our everyday lives. You interact with it constantly, probably without even realizing it. So, what do a burning mussel, a fluffy loaf of freshly baked bread, and a super trendy $30 bag of specialty coffee all have in common? Well, the answer isn't magic, and it actually isn't just basic chemistry either. It all comes down to a biological problem-solving mechanism. It's this incredible process that kicks in when a living cell desperately needs energy to survive, but finds itself completely, totally deprived of oxygen. To wrap our heads around how this works, here's our roadmap for today. We'll start with surviving without oxygen, then move into the lactic acid path, followed by the alcoholic path, hit on the anaerobic coffee craze, and finally wrap up with scaling up industrial fermenters. All right, section 1, surviving without oxygen. So anaerobic fermentation is basically a process that provides energy for cells without using a drop of oxygen. Usually, you see this in organisms living in oxygen-poor environments, or in simpler organisms that literally lack complex structures like mitochondria. For them, this all goes down right in the cytoplasm of the cell. It's basically their go-to way to extract energy from their food sources. Now, what's absolutely wild here is the stark contrast in efficiency. When a cell uses oxygen, aerobic respiration, it can squeeze out 36 ATP, which is basically the energy currency of the cell, from just a single molecule of glucose. But under anaerobic conditions, it cranks out only 2 ATP. Just 2! The cell is literally trading massive energy efficiency just to stay alive. It's essentially pulling the biological emergency brake. Here's exactly how that emergency protocol plays out. All cellular respiration starts with a step called glycolysis. This splits glucose into pyruvic acid, which yields that tiny little net gain of 2 ATP, and it reduces a molecule called NAD+ into NADH. Now, normally, with plenty of oxygen around, that pyruvic acid would cruise right along into a complex process called the Krebs cycle to make tons of energy. But without oxygen, bam, the process hits a brick wall. The cell is stuck with this pyruvic acid and literally has no way to make more ATP. So what happens to that pyruvic acid waste? Let's get into section 2, the lactic acid path. Well, the cell has to do something, because if it runs out of that NAD+, even that tiny trickle of 2 ATP from glycolysis is going to stop, and the cell will die. Game over. So it actually uses the pyruvic acid to oxidize the NADH back into NAD+. It basically just dumps its electrons right onto the pyruvic acid, transforming it into lactic acid. By doing this, the cell completely gives up on getting any more energy from the sugar. It focuses 100% of its effort on recycling NAD+, just to keep that basic glycolysis engine running. And here is where it gets delicious. We humans have basically weaponized this cellular workaround for thousands of years. Think about tiny bacteria, just 5 to 10 micrometers across, like lactobacillus. They perform this exact lactic acid fermentation. They digest the sugars in milk to give us yogurt and cheese, or the sugars in vegetables to give us kimchi and sauerkraut. So you know that signature tart, sour flavor you love? Yeah, that is literally the lactic acid waste product of a bacteria just trying to survive without oxygen. But hey, it's not just microscopic bacteria doing this. You do it too. If you've ever felt that intense, screaming muscle burn during a heavy lift or a crazy sprint, you are experiencing lactic acid fermentation firsthand. Your blood simply couldn't deliver oxygen to your muscles fast enough to keep up with the demand. Out of sheer desperation, your muscle cells temporarily switch to anaerobic fermentation. That burning sensation? That's just lactic acid building up in your tissue. Moving right along, let's check out section 3, the alcoholic path. So here we see a really cool divergence in the biological pathway. While humans and bacteria turn pyruvic acid into lactic acid, organisms like yeast and certain plants take a completely different route. When yeast is starved of oxygen, it takes that exact same pyruvic acid but converts it into ethyl alcohol and carbon dioxide. It has the exact same goal, recycling NAD+ to survive, but produces entirely different waste products. And let's be real, humans are absolute masters at harnessing this specific yeast fermentation. Think about bread, right? We mix yeast into dough, and as it ferments, we trap those little carbon dioxide bubbles to make the bread rise into a big fluffy loaf, while the tiny amount of alcohol just cooks off in the oven. For beer and champagne, we ferment sugars from grains or grapes, but we hold on to both the alcohol and the trapped CO2 for that amazing carbonation. And wine? We just capture the alcohol and let the CO2 escape. It's all the exact same cellular survival mechanism, just packaged differently. Now, let's pivot to something huge in the beverage world right now. Section 4, the anaerobic coffee craze. If you walk into a trendy cafe today, you're probably going to see anaerobic process stamped on a really expensive bag of beans. But here is the reality check. Anaerobic translates directly to without oxygen. And honestly, that's truly all we can accurately infer from the label. Calling a coffee anaerobic is kind of a catch-all buzzword in the industry right now. It doesn't tell you if specific yeast or bacteria were added. It could mean the coffee spent 24 hours in a basic plastic sack, or it could mean it sat for 10 days in highly engineered, specialized stainless steel tanks. To really see how this is done at the highest level, let's look at the La Chumeca micromill in Costa Rica, run by Martin Urania. His process is incredibly precise. First, he sorts the raw coffee cherries until they reach an exact sugar content, usually around 25 degrees of Brix. Then, he seals them away in specialized tanks for 48 to 72 hours. He's intentionally sparking this cellular fermentation process before ever taking them out to dry on tables like traditional natural coffee. But remember what we just learned about alcoholic fermentation? It produces a massive amount of carbon dioxide gas. So, as this coffee ferments inside these sealed tanks, the pressure builds up tremendously. To handle this, they use these really clever one-way valves. These valves allow the rapidly expanding carbon dioxide from the fruit's breakdown to vent out, which keeps the tanks from literally exploding, while completely restricting any outside oxygen from getting back in. And what's super fascinating is how they actually monitor this invisible process. They don't add any water to these tanks. Instead, the producers meticulously check the liquid and the Brix, the sugar content of the juices that naturally ooze out as the fruit cells break down in that oxygen-deprived environment. They track the health of the entire fermentation just by analyzing that oozing liquid way before pulling the coffee out to dry. Finally, let's zoom way out. Section 5, Scaling Up, Industrial Fermenters. When operating a massive industrial fermenter, engineers literally have to flip the script depending on the organism. Standard aerobic vats require vigorous air sparging, which is basically pumping oxygen into the mix, and they purposely leave airspace at the top of the tank. But for an anaerobic batch fermenter, you have to totally reverse standard procedures. There is absolutely no air sparging, and the tanks are filled entirely to the absolute brim, just to eliminate any dangerous airspace that might be hiding stray oxygen. Just look at the extreme measures they take to keep oxygen out of these giant vats. They blow nitrogen gas through the tank right at the start to purge any lingering oxygen. They pump the nutrient medium in hot and from the very bottom because hot liquids absorb way fewer gases. And get this, they barely even stir it. After an initial mix, they rely almost entirely on the natural violent bubbling of the carbon dioxide and hydrogen gases produced by the fermentation to stir the medium naturally. Oh, and they also toss in low redox compounds like cysteine just to be safe. The applications here are just staggering. By replicating this oxygen-free environment on a massive industrial scale, we are harnessing the desperate survival tactics of tiny cells to mass-produce vital stuff. I'm talking about using strictly anaerobic processes to churn out industrial chemicals like acetone, butanol, commercial lactic acid, and amazingly, even life-saving medical supplies like anti-tetanus toxoids. Which brings us to a final, pretty wild thought to leave you with. We've seen how a single, ancient biological workaround, a microscopic cell's absolute desperate attempt to survive without oxygen, has been manipulated by us to give us bread, wine, trendy coffee, and even life-saving vaccines. It really makes you wonder, what other invisible, microscopic miracles are happening around us right now, just waiting to be bottled up and engineered to shape the future of our food and industry? Think about that the next time you sip your morning coffee or feel that burn during a workout. Thanks for hanging out with me for this explainer, and keep exploring the incredible biology all around you.